Let us look at axis formation during fish development. Let us now switch gears to see how the dorsal ventral axis is formed in the fish embryos. Now, the embryonic shield plays an important role in the dorsal ventral axis formation. The shield gives rise to the dorsal mesoderm, which will form the notochord and somites, and it also causes the ectoderm to become neural tissues instead of epidermal tissues, similar to what we saw in the amphibian development. So, if we do an experiment where we take a portion of the embryonic shield, which is in the dorsal end of the embryo, and go ahead and transplant it into another embryo, but transplant it at the ventral end, then what we end up seeing is we actually see the development of two zebrafish embryos. So we see another embryo formed which has the anterior structures and the dorsal structures. And hence we can see the zebrafish that has two heads and two brains. Thus, the embryonic shield can actually help in the development of the dorsal ventral axis. Now, similar to amphibians, fish induce their epidermis by the actions of BMP and Wnt. And in the case of the neural development, BMP and Wnt is actually blocked by BMP inhibitors and Wnt inhibitors in the dorsal as well as anterior regions. And this is what we had seen in amphibians as well. Now, the shield corresponding to the dorsal blastoporlip gives rise to the dorsal mesoderm. However, it cannot give rise to the tail part of the embryo. And so this is where there's a slight difference between the fish and the amphibian embryogenesis. In the case of fish, there is a shield that's corresponding to the ventral blastoporlip, which is not as prominent as the dorsal part. Now, this ventral blastoporlip can actually give rise to the tail structure and hence is considered as a tail organizer. So this tail organizer in the figure is shown in the blue box as of the red structure. If that red structure, which is on the ventral side of the shield, is transplanted into an embryo and we allow the embryo to develop, what we observe is we can actually see the formation of a tail structure where this specific part of the donor was transplanted. Thus, the ventral blastoporlip or the ventral part of the shield is the tail organizer. Similarly, if we look at the cells that are making the lateral part of the shield, those cells give rise to the trunk and the posterior head structures. Thus, different cells present on the shield are actually able to induce the formation of different structures in embryos. Now let us look at what are the molecules that play a role in these processes. So in the case of the tail organizer, nodal and the bone morphogenetic protein play an important role. In fact, the ratio of BMP and nodal actually are the ones that play a role in forming the tail organizer. BMP tends to be highly expressed in the ventral margin, and the expression starts reducing as we go dorsolaterally and is very low in the dorsalmost region of the embryonic shield or the blastoporlip. And this is where we see the expression of nodal is very high. Now, if you remember, nodal is a member of the TGF beta family. Now we can see in the figure that is shown, we have an embryo where scientists have been able to manipulate the ratio of BMP and nodal in different parts of the embryo. And what we can see is when we have a gradient of BMP and nodal, we can actually stimulate the formation of tail structures in different parts of the embryo. That is what is seen in the top portion, the middle panel, where now the expression of nodal and BMP has changed its location in the embryo, and we can see that the tail formation, its axis is also different. And in the third panel, once again, when we have a change in the location of the nodal BMP expression, we can see that the tail axis has actually changed its direction. Thus, nodal and BMP play an important role in forming the tail organizer. Now, beta-catenin plays a role in establishing the nodal and the BMP gradients in the embryo. As we saw in amphibians, beta-catenin is highly expressed in the dorsal-most region. Let us now look at the anterior-posterior axis and the left-right axis formation. So when we look at the anterior-posterior axis, patterning of the neural ectoderm is similar to that observed in amphibians. Now, Wnt, FGF, and retinoic acid play roles in the formation of anterior-posterior axis. Similar to amphibians, Wnt is repressed in the anterior regions. However, Wnt, FGF, and retinoic acid are able to activate the posterior genes and hence help in the formation of the anterior-posterior axis. When we look at the left-right axis, once again there are many similarities to amphibians. In this case, nodal plays an important role in the left-right axis formation. Nodal target genes are important in instructing asymmetric organ migration and morphogenesis in the body. To summarize, we learned about the different axes that are formed during fish development and the different molecules that play a role in the formation of these axes.